This video abstract is AI generated. How does FTO know when to activate oxygen chemistry? FTO is a human iron 2 and alpha ketoglutarate dependent dioxygenase that removes methyl marks from nucleic acids. It is best known for demethylating M6A, an abundant RNA modification involved in gene regulation. And FTO activity has been linked to obesity, diabetes, and cancer. FTO catalysis requires two coupled reactions. In the secondary reaction, alpha ketoglutarate is decarboxylated and the iron 4 oxooxidant is formed. In the primary reaction, that oxidant removes the methyl group from the nucleic acid substrate, but this raises two key questions. Why does nucleic acid binding activate alpha ketoglutarate decarboxylation? And why are different methylated bases processed with different efficiencies? Activity assays provided the first clue. FTO demethylates M6A most efficiently, M1A more slowly, and M3T very poorly under the tested conditions. This means that the identity of the methylated base strongly controls catalytic turnover. Solution NMR then showed that substrate binding shifts the FTO/alpha ketoglutarate conformational equilibrium methyltrosy chemical shift perturbations were largest for the M6A containing DNA, intermediate for M1A, and smallest for M3T. Importantly, the magnitude of the NMR shift correlated with catalytic activity. Molecular dynamics simulations explained the structural origin of this effect. Alpha ketoglutarate can occupy two metal-centered conformations. In the offline conformation, alpha ketoglutarate keeps the iron 2 center poorly accessible to oxygen. In the inline conformation, alpha ketoglutarate rearranges so that the metal becomes exposed and the future iron 4 oxooxidant is properly aligned with a methylated base. The methylated base acts as the trigger. Bulky methylated bases sterically push alpha ketoglutarate toward the inline state. M6A provides the strongest shift. M1A gives an intermediate effect, and M3T fails to efficiently stabilize the inline conformation. This switch controls oxygen activation. Only the inline alpha ketoglutarate state efficiently exposes iron 2 to incoming oxygen, allowing formation of the reactive iron 4 oxo species needed for demethylation. The final model is that substrate binding activates FTO by shifting alpha ketoglutarate inline. This exposes the metal to oxygen, couples oxygen activation to the presence of the correct methylated substrate, and explains why M6A is demethylated more efficiently than M1A or M3T. To know more, read our work online.